Thank you, Justin, for this nice introduction. Hello, everybody. Thank you for joining us tonight for this webinar on canine reproduction. You heard the South African accent of Justin. Now you're going to hear my French accent because, as he told you, I'm from France. And I'm very happy to be there tonight lecturing for you on the web on this very interesting topic which is timing of ovulation in the canine species. Because there are lots of techniques that are available today for timing of ovulation. But think about it. When I was preparing this talk, I thought, oh, that's going to be very interesting for me because when you speak about timing of ovulation to normal people, imagine yourself walking on a very large avenue. I think we should take a very big avenue to do those kind of experiments and speaking with normal people, asking them, what do you think of timing of ovulation in the beach? And this is an experiment I did in the past. And these are the kind of answers I got from people. What? Seriously? Or something like, you're kidding me, right? Where is the camera? Because again, timing of ovulation is something very specific. And when we speak to people that don't necessarily have the understanding of what canine breeding involves, this might be something they really don't understand why we're doing that. And that's why I was really happy to do this webinar tonight, because I thought, okay, I'm going to speak about something I worked on during several years to passionate people who clearly understand the value and the importance of a service like timing of ovulation. Because you know that, well, canine fertility usually is not bad. I'm not going to tell you bullshit. Canine fertility is usually excellent, quite good, especially if we compare it to human. But when we breed dogs, when you guys breed dogs, your goal, what you want is to improve the breed. You're passionate about what you're doing, and you build your genetic selection program in order to bring the breed you're passionate about to the next level. And fertility sometimes can be tricky. If we do the basic maths in terms of fertility, it shouldn't be that complicated, in fact, when we think about it, because we need oocytes, spermatozoas, which meet at the appropriate time, and if they meet at the appropriate time, fertilization will occur, and in the end, we should have puppies. That's the basic maths. And when we look at it like that, we can think, oh, it doesn't seem that difficult. Why do we need a timing of ovulation? Because there are several parameters we need to focus on concerning those gametes. For the spermatozoas, for instance, for sure we need them to be there. But more than that, we need them to be numerous. We know that today, to ensure fertility in a bitch, we need at least 150 million motile sperm in the ejaculate to ensure pregnancy. If you have less than that, maybe the bitch will not get pregnant just because there's not enough sperm to fertilize this bitch. By the way, keep in mind that usually this number is quite easy to reach because a 1.5 kilogram Yorkshire Terrier will produce approximately 250 to 300 million sperm in its ejaculate. But when dogs get older, this can be a critical factor, a limiting factor. We need them to be normal. In an ejaculate, one dog's ejaculate, it's not abnormal to see abnormal forms, I should say. There are always some spermatozoa which don't have the proper shape, the correct shape to fertilize the oocytes, but there shouldn't be too much of them. If we have more than 40-50% abnormal sperm in the ejaculate, this is when it can impact fertility. And fortunately, this is something we can assess by doing a spermogram in the male dog. Something else important, we need them to be motile for sure. Motility is the most important parameter when it comes to fertility in male dogs, because if you have lots of sperm but none of them are moving, no, you will not get anything out of this breeding. So you see that spermatozoa, some criteria are essential to ensure fertility. And same thing for oocytes. Oocytes, the eggs which are produced by the female. We need lots of them. They need to be numerous. I was recently speaking with one of our breeder partners, and I was telling them, oh, the largest litter I've ever had in a bitch I've ever seen was 22 puppies in a Basset Hound. And they said, oh, I saw 24 in a Great Dane. So you see that the limiting factor when it comes to the litter size is the number of oocytes ovulated by the bitch, and this varies a lot. Something else to focus on is that those oocytes needs to be normal. We know that when bitches get older, the morphology of the oocytes might change and might not allow fertilization, proper fertilization of these oocytes. And something extremely important, and that's the purpose of timing of ovulation, is that we need fertilizable oocytes. We need those oocytes to be able to accept fertilization. We need those oocytes to accept the sperm cell to penetrate the zona pellucida and start the first steps of embryonic development that will lead them to fetuses and then to the puppies as we expect. So plenty of parameters to focus on those two different gametes. And in the end, what really matters is that when we have lots of good quality sperm cells in the oviduct, because this is where the fertilization will occur, we also need as many fertilizable oocytes as possible. And these are the basic maths we try to solve when we're breeding ducks. And you guys know that you're breeding a very, very, very unique species when it comes to reproductive physiology. We can think that dogs are mammals and they should work the same way like, I don't know, cows, mares, sows, women, etc. But in fact, it's absolutely not the case. They are very unique in terms of reproductive physiology. They have very unique features. In most mammals, when ovulation occurs, typically, as soon as ovulation occurs, the oocytes that are released in the oviduct are mature. If there are some spermatozoa around, those spermatozoa can fertilize this mature oocyte. And that's why maybe you're familiar with bovine or equine, but in bovine and equine, very often when we need to breed them, ovulation, we need to breed them very close from ovulation. Maybe even sometimes they will be bred before ovulation, just before, to be sure that fertilization will occur. So in this species, it's a tricky part to determine when ovulation occurs because we have a very, very short window of fertility. In canines, it's totally different. In canines, they will ovulate, but the oocytes will release premature oocytes. And those premature oocytes, they will take 56 to 72 hours to become mature, to be able to accept fertilization. This is a very unique characteristic in terms of reproductive physiology. This makes timing of ovulation somehow easier, and it's something very interesting for you guys when you breed dogs, because if you think about it, you need to detect ovulation, and then you have 2-3 days to go breed the bitch. So you don't need to breed them very close to ovulation. It's not like they ovulate and you need to breed them as soon as possible. You have time, and that's a great advantage in order to time the breedings. But for that, you need to know when ovulation occurs. Because those oocytes, they are mature, but when they're mature, they will survive 48 to 72 hours. And then they will degenerate. And when they degenerate, they are no more able to accept fertilization. They are no more able to be fertilized. And because of that, that's why if you breed them, the bitches too late, they will not get pregnant. So you see, the maths look easy, but in fact, it's a real challenge. Because if we think about it, the estrus period, the seasons of the bitch, lasts usually approximately 3 weeks. The fertility window, however, lasts 2 to 4 days. So we need to target a 2 to 4 days fertility window on a 3 week season. Very small window, very easy to miss, as you can imagine. And we're going to break the neck to some of the most common misconceptions I've seen in this field. Very often when it comes to breeding, and I'm sure you guys heard that in the field, we speak a lot about the ability of the male to detect ovulation in the bitch. And don't get me wrong, certain males, they're able to smell, to detect some pheromones that we are not able to perceive, and we don't know how to interpret them today, that might be well correlated with the ovulation in the bitch. And for sure, there are some males which know when to breed the bitch. However, That's why, by the way, we use dogs. Dogs are able to detect ovulation in cows. Some dogs are able to detect ovulation in cows. Some dogs are able to detect cancers in humans. However, I would never rely only on the male because it's really individual dependent. Think about it. I've seen some males, and I'm sure you guys saw similar things in the field, where they are interested in any female. They would mate any female that come close to them. Well, I've seen some males which were only interested once in a female. I remember a boxer male dog. He only wanted to mate a female once, and then he wasn't showing any interest to this female at all, even if she was in an optimal fertility window. So the male factor is not always as accurate as it seems, and it's very difficult to be sure that the male always bred at the right time. So don't trust too much the male, because sometimes they can make mistakes and they are not as efficient as we might think. Same thing for the females. I've heard in the field that sometimes the female will only accept the male when she's ready to be bred. Maybe that's the case for some of them, but not all of them. If you're breeding some very dominant bitches, for instance, if you breed some American Akitas or some Chow Chows, very often if you let the male and the female together for breeding, maybe the female will just try to kill the male because she's so dominant that she doesn't want the male to mount her. And that's, again, you know your animals, you know they will react, and maybe you will be able to detect those bad behaviors. But the female not accepting the male doesn't mean that the female is not ready to be bred. Unfortunately, it's just a behavior that is very difficult to interpret. And the most important thing, the biggest misconception out there, is in the books, in the old books, I would say. When I was a vet student 10 years ago, well, even more than that, 13 years ago, I remember sitting in this classroom and I was listening to my teacher in animal reproduction. It was a guy who was his main interest was in bovine reproduction. He told us that he was teaching us canine reproduction, but he had no interest at all in it. And he said something like, bitches are always ready to be bred between the 12th and the 50th day after the beginning of the season. And you know that it's not always true. There's a story I like to tell. Few years ago, I had this German Shepherd bitch coming for a timing of ovulation. This bitch came to us 6 days after the beginning of the season. She was supposed to be bred in Germany. And the owner said, okay, we have time. I come a little bit early, but I just want to be sure. I don't want to miss the day of ovulation. I don't want to miss it. We did the timing of ovulation in this bitch. And interestingly, on the 6th day after beginning of the season, she had already ovulated. So he had to rush to drive fast to Germany to breed the bitch. And fortunately, 3 weeks after, when he came back for a pregnancy diagnosis, the bitch was pregnant. And she had 8 puppies in the end. Great story. 1 year and a half after, this same owner came back with the same bitch. And he said, this time I really don't want to miss the bitch. I will come as early as 4 days after the beginning of the season. We followed this bitch until the 30th day of Eats. She ovulated on the 30th day of Eats. She was bred on the 32nd day of Eats. She got 12 puppies. So same bitch, 2 different estrus cycles, 2 different ovulation dates, and as you can see in both cases, if this bitch had been bred between the 12th and 50th day after beginning of the season, she might not have conceived. Sperm can live at least in average 2 to 4 days in the genital tract of the bitch, fresh sperm, but in case the semen quality is not optimal or whatever happens, in this case you can see that once she ovulated at 6 days, the other case in 30 days, if we had bred her between 12 and 15, she wouldn't have get pregnant. So there are lots of variation from one bitch to the other, from one cycle to the other. And the only solution, if you want to optimize the fertility of your animals, is the timing of ovulation. And today the good news is that we have many different techniques. Some of them are excellent in order to detect ovulation and to help you solve the challenge that you have to face when it comes to canine reproduction. Again, as I said, you guys are passionate, you have your breeding program and you want to bring the breed to the next level by doing your genetic selection program. And the best way to optimize it is through this ovulation timing, because by doing that, you ensure, well, you give all the chances that the bitch will be fertile, and you have all the chances that you will have a larger litter size. You will optimize prolificity of the in this bitch, because in the end, that's also what matters. If you want to, the more subjects you can have from a specific breed, the more valuable and the more interesting your genetic selection program can be. So there are many techniques, as I said, and which one to pick? Because this is the question. There are so many of them. What can we say today in 2014 about timing of ovulation in the bitch? There are some hormonal assays that can be used. Some of them focus on the vaginal secretions of the bitch during their seasons, and some of them are based on visualization. And this is a very exciting field of veterinary medicine that recently became available to the veterinary medicine, and that offers us great opportunities in terms of timing of ovulation. So let's speak a little bit on hormonal assays in the bitch. And if you ever attended to a seminar of teriogenologists like me, I'm sure you already saw this picture, which is in fact a graph from an article of Pope of Canine, well, the Pope of Canine Reproduction, Pat Concannon. This article was published in 1988. It shows you the different profiles, the different hormonal profiles of the bitch throughout all the different reproductive cycles. And tonight we're mainly going to focus on what happens during our seasons, during estrus, proestrus and estrus, because this is when we will need to focus on the animal to detect when ovulation occurs. I'm sure you heard about progesterone assays, and progesterone assays are just clearly for me the gold standard to detect ovulation today in bitches. Why do we use progesterone assays in bitches? Why don't we use it in other species? For a simple reason. Told you, canines are very unique in terms of reproductive physiology. And if you look at this picture here, which is an ovary of a bitch just before ovulation, you might see those big round-shaped bubbles on the surface of the ovary. These are ovarian follicles. And those ovarian follicles have one specificity in the bitch. In all other mammals, they secrete progesterone after ovulation. In fact, they ovulate, they transform, those follicles are transformed in a structure we call the corpora lutea, and this corpora lutea will produce progesterone. And this is when we can assay progesterone in most mammals. In the bitch, those ovarian follicles will secrete progesterone before ovulation, which means that when we look at the curve in terms of progesterone, this rise of progesterone will be detected before ovulation. And that's why we can use this technique to determine the day of ovulation in the bitch, while we are not using it in the other species, because in the other species it will tell you that ovulation occurred. In the bitch, it tells you when ovulation occurs. And to understand that, I just wanted to show you this graph. This is a study we did in 2004. And it's very interesting. The first thing you can see here is that there are different, all those graphs represent different progesterone profiles of different bitches. As you can see, they are highly variable from one bitch to the other. And what I can tell you after doing more than 7,000 timings of ovulation using progesterone assay in a bitch is that you can have many different progesterone profiles from one bitch to the other, and even from one cycle to the other. But basically, us teriogenologists, we focus on three essential points on these curves. The first one usually is around a progesterone value of 2 to 3 nanogram per ml. 2 to 3 nanogram per ml usually corresponds to the LH peak. LH is an hormone that is secreted by the brain of the bitch, and we'll talk about LH later, but LH is usually it's a pulse that will trigger ovulation. It's a triggering signal for ovulation. And usually this LH peak is reached around 2 to 3 nanogram per ml. And this is very interesting for us to know about that, to have an estimation of the LH peak, because we know that from the LH peak to ovulation, there's approximately 48 hours. And then the second point we will focus on is ovulation. You can take a 1.5 kilogram Chihuahua, you can take a 120 kilogram Bull Mastiff. All of them will usually ovulate at the same progesterone value. They usually ovulate around 5 to 6 nanogram per ml of progesterone. And that's very consistent. Whatever the bitch, the value of ovulation, and that has been proven by many, many different studies, is approximately around 5 to 6 nanogram per ml. And again, you can take a large bitch, a small bitch. This is very consistent. So we know today in the bitch, the progesterone value at ovulation. This is something we can determine. And this is something that is clearly demonstrated by many, many scientific publications. However, look at the curve after. 2-3 days after ovulation, which means during the optimal fertility window. Look at the curves. They are very distant from one another. And that's important because sometimes I meet people that and they're telling me, oh, you know, the best time to breed the bitch is when the bitch reaches 25 nanogram per ml, or 35 nanogram per ml, or 40 nanogram per ml. That doesn't mean anything. 2-3 days after ovulation, the bitch can be at 10, 20, 30, 40, above 60 nanogram per ml. It doesn't mean anything. It just tells you that the bitch ovulated. When? If you don't have a previous progesterone value to extrapolate and try to estimate the day of ovulation, you won't be able to tell if you just have this high progesterone value. But there's no perfect level concerning the fertility window. We know we know at which rate of progesterone the bitches will ovulate. We don't know what is the best rate to breed them. Again, I said sometime I followed some Afghan hoons, and in Afghan hoons, very often 2 days post-ovulation, they are above 16 nanogram per ml in progesterone. And we bred them, and they were fertile, and they had puppies. So just because it was 2 days post-ovulation, why do we need this rate? Why do we need this extra progesterone rate if we just need the 6 nanogram per ml to detect ovulation? For a simple reason. If 2 days after ovulation, the progesterone should be high. If it's not high, if it does a plateau, if it's still at 5 or 6, etc., it shows you that maybe there is something blocking ovulation. Maybe, I don't know, there is an ovarian cyst. Maybe the bitch is doing an anovulatory cycle, which is extremely rare usually when they eat 5 to 6 nanogram per ml. Or maybe they're doing a luteal failure. So that's why we need this third progesterone rate, this high value, just to be sure to confirm ovulation and to confirm that progesterone is rising. And if you listen to what I just said, you may have noticed that I spoke in nanogram per ml. And that's something important too, because sometimes I have clients calling me, telling me, oh, the bitch is at 2. What do you think, doc? And 2, my question was 2 what? Because for sure, sometimes the unit that is used is nanogram per ml, but sometimes, or certain machines, certain country, will use nanomole per liter, which by the way should be the official unit. And 2 nanogram per ml is usually around, corresponds approximately to the LH peak. 2 nanomole per liter is a very, very low rate, so we will not interpret the 2 results in the same way. So the unit is important to know, and I showed you here on this slide, or we can move from one unit to the other, but keep in mind that when you speak to somebody about progesterone rates, you need to mention the unit that you're using, because depending on the unit, we will interpret totally differently the progesterone values. Again, I said that on ovulation is at around 5 to 6 nanogram per ml, but if it was nanomole per liter, it should be between 15 to 20 nanomole per liter. And I mentioned those values of ovulation, etc., and I must admit it's not totally correct for one simple reason. The 5 to 6 nanogram per ml I mentioned were things I obtained on my machine at the vet school, the machine I was using, and we were what we call a reference center for progesterone testing, so I knew that on this machine, between 5 to 6 nanogram per ml, ovulation was occurring in the bitch. But I recently had a discussion with a veterinarian in Western Canada, and we discussed about the difference between machines, and for sure, if you use a different machine, you can have a different result. let me explain you why. Those machines are human machines because progesterone in all mammals is the same. So usually we use machines for humans, and in France, for instance, we often use human labs to assay progesterone. I know it's not always feasible in Canada and the US, but in France that was an option we had, and that was a very good way for our breeders to go to their own human lab and to do their progesterone testing with them in partnership with their veterinarian. So, but the thing is that those machines, in humans, we want to know if there is a lot of progesterone or not, and if there is progesterone or not. That's the only thing we need to know. So they don't need to be as precise, as accurate as we are in dogs and cats. So 5 to 6 nanogram per ml on my machine could have been 8 nanogram per ml on another machine, or 4 nanogram per ml on another machine. So when you assay, for instance, if you use this machine, which is a mini Vidas, which is one of the machines that is very often found in private practice when I was working outside of the vet school, very often this is the machine I was using inside the vet clinic. And for instance, I remember once I was using this machine, I had a result, but I couldn't interpret the result. I need to take this result and I needed to put this result into an equation so I could convert it in the value I would have obtained with my machine in the reference center, and so I could interpret it. So just to tell you that more than the machine, what is more important in the interpretation. And you need somebody who is used to the machine and who is used to interpret the value of this machine to benefit from this testing. I just wanted to show you this small machine as well, which has been, I recently discovered it. It's a small machine that can be used in-house at the vet clinic now, which is really affordable and which provides you results very quickly. And which provides you with quantitative progesterone results. So like the one we have on these two machines. And this is now an option that veterinarians who are doing lots of timing of ovulation can try. And if they are comfortable with the interpretation on this machine, that could be a very interesting tool for you guys, especially if you have a breeding during the weekend. I often hear people complaining about the fact that, oh, you know, we have to breed this bitch and it's during the weekend and we have to ship this blood sample to the lab, but we will only get the result maybe on Monday or Tuesday, etc. Today we have in-house machines which are affordable and that veterinarians can use. So if you work a lot with your veterinarian and if they do lots of progesterone testings for you guys, this could be something you could discuss with them. Those machines are available and they could be something interesting for them to have in the clinic. Then for sure it depends on the activity they have in canine repro. But this is something you can bring to their attention, that there are some machines that give you quantitative progesterone assays and that are affordable today. And I was speaking about quantitative progesterone assays, which are those assays you do and you get a value. They will tell you the bitch is at 2 nanogram per ml or at 4 nanogram per ml. There are also semi-quantitative assays, which are like this one. It's a colorimetric reaction. You take the serum, the plasma of the bitch, you put it in those assays, in those cups, and there will be a colorimetric reaction. We'll evaluate this colorimetric reaction, and it will tell us, depending on the color we obtain, if the bitch is below 2 nanogram per ml, between 2 and 4 nanogram per ml, between 4 and 10 nanogram per ml, above 10 nanogram per ml. This will give you an estimation of the day of ovulation. This will give you an estimation of the progesterone rate, and we have different type of tests that are available today. I know at least 3 of them. We can even use the machine I just showed you, which is, as I said, able to do quantitative progesterone tests, but which is also able to evaluate, to better evaluate, I would say, those semi-quantitative reactions, because sometimes the color can be tricky and we could misinterpret the color. With this machine now, they said that it might be easier, it might increase the efficiency of interpretation of those tests. So what I want to say is that we have options that can be used in-house at the vet clinic now. And those, if your vet doesn't do lots of quantitative progesterone tests, maybe if you need to do a timing of ovulation, you can discuss with them so that they can bring in some semi-quantitative tests, so that they can work with these semi-quantitative tests just for this timing of ovulation. So there are options. progesterone assays are available today, and there are options for you guys to use them in partnership with your veterinarian. So don't hesitate to bring this to their attention if they are not familiar with the technique. However, you see that we have two different kinds of progesterone assays, and they are great tools. But depending on the type of artificial insemination, the type of breeding you would like to do, we cannot always use both of them. For natural breeding, fresh semen artificial insemination, for sure, quantitative progesterone and semi-quantitative progesterone are okay. Keep in mind that semi-quantitative progesterone assays gives you an estimation of the ovulation date, but sperm cells, fresh sperm usually lives up 2 to 4 days in the genital tract of the bitch. So in the end, even if you breed the bitch a little bit earlier, that should still be okay with fresh semen. So semi-quantitative progesterone assays are great for fresh semen artificial insemination. In terms of shield semen artificial insemination, however, it's not the same, because again, well, we could theoretically use semi-quantitative progesterone assays for shield semen AI. However, we're dealing with a semen that has a shorter lifespan than fresh semen. So it's an available option, but if we want to have all the chances, to put all the chances on our side, I would definitely prefer to go for quantitative progesterone assay. And if you do frozen semen AI, then you have no choice to move to quantitative progesterone, because frozen semen lives up to 4 to 6 hours in the genital tract of the bitch. 4 to 6 hours. You can't use only an estimation of the moment of ovulation. You need to be extremely accurate, and for that, definitely better to move to quantitative progesterone assays. I would never recommend to use semi-quantitative tests for frozen semen insemination. And just to show you, this is the result we got in our facility on 532 bitches. But as you can see, if we have good timing, we have good results. 73.5% fertility rate using progesterone assays is excellent, especially when you think that many of those bitches that were coming to see us were coming for infertility at the beginning. So great fertility rate can be obtained if we do a good timing. And look at the litter size. In average, 6.2 puppies per liter. So, as I said in the beginning, progesterone assays, hormonal assays like that will help you optimize the fertility, but it will also help you to optimize the prolificity of the litters you can have by using those tests. And there are other hormones we can focus on when it comes to timing of ovulation. And the other one we will not focus on is the LH. And when it comes to the LH, we're looking for the peak. As I said previously, LH, when you do progesterone assays, you're looking for a steady increase of progesterone secretion. And you might have to do maybe 2, 3 progesterone assays. You will see it increasing, you will reach the ovulation value, and when you know when ovulation occurs, you are able to schedule the breedings. With LH, which again triggers ovulation, it's a little bit different. The secretion of LH is pulsatile, which means that the concentration varies a lot throughout the day, and what we want is to find out when this LH peak occurs, because from the LH peak to ovulation, we know that there is indeed 48 hours. And if we know when the LH peak occurs, we know when ovulation will occur, and again, we can schedule the breeding events. But this LH peak lasts 12 to 96 hours. So for sure, if you're in a case when it lasts 96 hours, you might be able to find it out. If you're in a case where it lasts 12 hours, it might be a little bit more tricky. As I showed you, progesterone, when you're getting used to do the timing, sometimes you can do one progesterone assay every 2 days or every 3 days. You can interpret the value of the progesterone and then determine when you will see the bitch for performing another assay. And again, as I said before, usually when we were doing timing of ovulation using progesterone assays, most of the time we were doing 3 of them to be able to clearly detect the day of ovulation. With LH, since it's a pulsatile secretion, we need to do it on a daily basis. And look here, for instance, if we're dealing with a 12-hour LH peak, even if we're doing it once a day, we could miss it. In the past, we were using quantitative LH assays. And to do that, we had to do 2 LH assays per day. every day to detect ovulation. Very expensive, and today it's mainly done in research facilities. But maybe you have heard of semi-quantitative LH tests that are now available, which again gives you an estimation of the LH concentration. But what you're really focusing on here, you just want to see the peak. You just want to see if this value increases. And when you see the peak, this is when the test turns positive. You know when the LH peak occurred, and you can determine when to breed the bitch again. And recently there was this publication, this article published presented at a vet seminar 2 years ago, where they only used LH kits, LH tests, semi-quantitative LH tests to determine the day of ovulation in 10 beagle bitches. So they determined the day of ovulation by doing daily LH assays, and they bred the bitch according to the day of ovulation based on the LH test. They took they split the sample so that they could perform progesterone assays at the same time, but blinded, so nobody could have access to the true values. And the 10 bitches that were bred were found pregnant. So this shows you that semi-quantitative LH tests can be used alone to determine when is the best time to bred the bitch. However, we need to do it on a daily basis, and that's why many of my colleagues, in fact, are using it today in their vet clinics, but they're using it retrospectively. They do the progesterone, they determine when ovulation occurs, and they keep those frozen samples, they keep the serum frozen, and then they evaluate the LH on these samples they took to assay progesterone as well. So then they know when ovulation occurs, and they go back to try to find out when exactly the LH peak will occur. And that's a very interesting approach, especially when you're dealing with frozen semen insemination, because again, you want to be extremely accurate. But as you can see, the advantage with LH, semi-quantitative LH tests, is that they are available in-house at the clinic. It doesn't take long to get the result. So that could also be an interesting alternative depending on where you are, on how easy it is to access progesterone assays to do a timing of ovulation. And to sum up on these hormonal assays, clearly these tests are it's usually important to perform them at the vet clinic. And I showed you the result we could have with good timing with progesterone assays. So if you have a great partnership with your veterinarian, you can have great results in terms of timing of ovulation thanks to this technique, which are today, again, easier to access in-house at the vet clinic. So don't hesitate to bring this to the attention of your veterinarian and to discuss this with them, because there are alternatives today that they can use in their clinic to help you find out what is the best moment to determine the day of ovulation in your bitch. So these are the hormonal assays, and as I said, these are, for me, the gold standard today in veterinary medicine. There are other things we can focus on, especially the vaginal secretion in the bitch. And the first thing when I speak about vaginal secretion, the first thing we usually focus on are the vaginal smears. So the vaginal smears are quite easy to perform. Usually we will insert a vaginal swab in the vagina of the bitch. You can see here that we can, remember the vagina of the bitch is 15 to 30 centimeter length, so we can insert this swab very far in the vagina to be sure to collect cells from the vagina, and we'll roll this swab on a slide and we'll interpret it under the microscope. And what we will evaluate is the morphology of this cell, the aspect, the shape of these cells, and based on this evaluation, we'll be able to tell you at which stage of the estrous cycle the bitch is. And why is it important? Just one quick reminder: the vaginal wall of the vaginal wall in the bitch, its morphology will change during estrus, during the season, because of hormonal variation of the hormonal secretion, especially the estrogens. When the bitch is in an estrus outside the breeding season with no hormonal impregnation at all, this is what the vaginal wall looks like. It's very thin, few layers of cells, and typically when we do vaginal swabs, we'll see those small, round-shaped cells with big nucleus that shows you that there is no hormonal impregnation at all. Then the bitch starts her season. Because of the estrogens, the vaginal cell starts to differentiate. and they become bigger. They become more angulous. And when you do the vaginal smears, you should see something like that. So you see the morphology is totally different. They are more angulous, the nucleus is smaller, and when we see that, we know that, okay, she's in estrus, she's in proestrus, she's at the beginning of her estrus cycle. And then comes the estrus. This is when we have the maximal differentiation of those cells. Those cells then, they look like what we call complex. They are very angulous, a very small nucleus, and when we see pictures like that, we can tell you that the bitch is in estrus, which means that this is the period which surrounds ovulation. We know that when we have this vaginal smear, ovulation will occur during this period. And then we have the diastrus smear, because after ovulation, progesterone rises, we have a high level of progesterone, and the cells, they differentiate them, very hard to say in French, and they turn back to this round-shaped aspect, and this is what we will see. What does those vaginal smears tell us? The first two ones, when we see them, we can tell you that it is too early to breed the bitch. The last one, when we see it, we know it's too late. And usually when we see that, we know that it's pointless to breed the bitch because we are at least 7-10 days after ovulation. The cervix might be closed, and this is definitely not, we moved away from the optimal breeding window. It's too late, it's pointless to breed the bitch if we see that. And when we think about this, in fact, this is the one we should look at. If we see that, we should say, okay, we have this estrus vaginal smear. This is the period surrounding ovulation. When we see that, this is when we should breed the bitch. And to tell you the truth, in the past, many of my vet colleagues were doing that. They were doing a vaginal smear. When they saw this estrus smear, they were telling the client, okay, now you can breed the bitch. But we know today that this estrus smear, we can have it from 5 to 10 days before ovulation to 5 to 10 days after ovulation. So again, it's a great technique to detect the estrus, which is again the period when ovulation will occur. And to tell you the truth, today at the vet clinic, we were mainly using it in combination with progesterone assays to determine when to start doing progesterone assays. Typically, when we were seeing those proestrus smears, we knew that it was too early, and we were not starting the progesterone assays. We were only starting them when the vaginal smear looked like estrus, because we knew that we were getting closer to the ovulation period. And the combination of two could be a very interesting way to decrease the amount of progesterone rates that are required to determine the day of ovulation, and it could be a cost-effective method for you guys as well. So as you can see, it can. I was still doing vaginal smears each time we were seeing a bitch at the vet clinic, because I think it brings us lots of information that we can combine with the progesterone assay and that we can interpret. And one I was very interested in was this one. If you look at this smear, and if you look, those structures I circled here, these are neutrophiles. Those neutrophiles show you, when you see them, especially on an estrus smear or on a vaginal smear, that there is a genital inflammation. If you see that, especially when you're doing a timing of ovulation, you know that there's something wrong in the genital tract of the bitch. Could be a vaginitis, an inflammation of the vagina. Could be, let's be crazy, a pyometra. It could be an endometritis, an inflammation of the endometrium of the bitch. When you see that, you know that there's something wrong. And you know that if you breed the bitch, because sometimes you don't see purulent vulvar discharge, you don't see those obvious signs of vaginitis, you just have this finding on the vaginal smears. When you see that, you know there's something wrong. And if you see that there's something wrong, you know that you have to take other steps. You have to add some treatment to be sure that the bitch might be fertile. And what do you think of that? Look at this picture. And this is something I had, it's a discussion I had recently with one of our breeders. She was telling me about something called ferning. And this is exactly what ferning is all about. Ferning, typically, the vaginal mucus during estrus in the bitch, if you collect it and you put it on a slide and you let it air dry, it will adopt a crystallization pattern, which is something like that. And these crystallization patterns become more and more complex. And when it reaches its peak in terms of complexity, this is usually when ovulation occurs. And then you will see it becoming less and less complex. after ovulation. So this is basically what ferning is about, visualizing those this arborization pattern, this crystallization pattern, in order to determine the day of ovulation. And there are some studies that were done in the beach, and as you can see, this one was done in 1989. So they evaluate the crystallization pattern in the vaginal mucus of the beach. And at this time, keep in mind, it wasn't easy to access progesterone assays. So that's why they were thinking of using this ferning pattern in correlation with vaginal smears in order to better evaluate the day of breeding. And what they found out is that, okay, from the LH peak to the ferning peak, because again, remember, we want to visualize when we get this highly arborized, highly complex ferning pattern. When we were from the LH peak to the ferning peak, well, there were approximately 2 days, which means that in fact, the ferning peak approximately corresponds to the day of ovulation or 1 day prior to ovulation. But look at the extremes. Sometimes this ferning peak was seen 2 days before the LH peak, which means approximately 4 to 5 days before ovulation. Sometimes it was seen 8 days post the LH peak, which means approximately 9 to 10 days post ovulation. So it was an interesting tool, but it's extremely complex because remember, you need a lot of experience to evaluate how complex the ferning pattern is. And as you can see, there are lots of individual variations. So that's certainly why this technique today is nearly never used in most of the canine breeding centers that I know about, because unfortunately it's not as accurate as the technique we have today. Could be a valuable tool, but in certain beaches you can see that there are lots of variation and it might not fit to all situations. And this is an interesting part, because maybe you heard about those ovulation test strips, and I must admit I've never heard about it before I came to North America. Very interesting, because in fact those ovulation strips, if I'm correct, usually you put them in the vagina of the bitch, and you have this pink color that will turn into another color and when you see this modification, it tells you when the LH peak is. And then, because you know when the LH peak occurred, you can define when to do the breedings. And you have to do this on a daily basis. This is basically what is recommended. These strips, in fact, they detect a modification of the glucose concentration in the vagina of the bitch. And this is something that was described in 1973, so long time ago. And honestly, when I heard about this technique, I was wondering, okay, I spend lots of hours learning about the techniques that are used in canine reproduction to sit for the exam to become a specialist in canine and feline repro, but I've never heard about this technique. And I looked at this article, and what they said is that although initial results were promising, this technique has not found clinical acceptance, presumably because of individual bitch variations. There are very few data on this technique, and again, maybe they are interesting in certain cases, but from what we know from the scientific literature, there are lots of variation, and it's not the most reliable technique out there that you can use to detect the day of ovulation in the bitch. What about resistivity of the vaginal mucus? I don't know if you ever heard about this technique, but as you can see on this picture, it's basically a probe that we will put in the vagina of the bitch that will measure the electrical resistance of the vagina of the bitch. And based on this measure, we could evaluate when ovulation occurs. Because in fact, the electrical resistance of in the vagina of the bitch is influenced by the hormonal secretion, the hormonal modification that happened during the season of the bitch. And we should usually obtain a curve like that. So it starts to increase at the beginning of estrus, during proestrus. It reaches a kind of peak, and then it starts to decrease. And when you see this decrease, this is when ovulation occurs. So there are no perfect number or value of resistance that will tell you when ovulation occurs. But if you see this pattern, this increase and then this drop, when you see the drop, this is usually when ovulation occurs. And again, it has to be done daily. This technique was evaluated, I think, The first publications were in the 80s, and the conclusions were that this technique was as accurate as a vaginal smear. And in 2009, one of these devices was sold in France. And one of our students said, okay, what about this? And we said, maybe we should have a look at it. And we did a study on 26 beagle bitches. And on these 26 beagle bitches, we measured the resistivity of the vaginal mucus on a daily basis, and we compared it to progesterone assays. And what we found is that when we saw this decrease, which usually is when ovulation occurs, this decrease occurs 24 hours around ovulation in 73% of the cases. So it was a good way to detect ovulation in approximately more than 70% of the animals. Keep in mind, this study was done in beagles, and in one of them, this decrease was detected 60 hours post-ovulation, which is still not that bad when you think about it, because it could be an interesting tool to use if you do a fresh semen insemination, for instance. However, these were done in beagles, as I said. So what we learned from this study is that depending on where you put the probe in the vagina of the bitch, the measures are totally different. So you need to be sure when you do the measurements on a daily basis that the probe is always at the exact same place. And this is in beagles. I used to say when we were doing some experiment at the vet school that when we do something in beagles, everything works in beagles. Maybe it's different in Labradors, maybe it's different in Rottweilers, maybe it's different in German Shepherd or Yorkies. Hard to say. Unfortunately, we don't have enough data. It could be a very interesting tool, but it needs to be evaluated for your specific situation and for your specific individuals. I knew a breeder in north of France who was using this technique, and I remember discussing with her, and I said, what do you think of this technique? And she said, well, it's quite accurate. Some bitches misses, but most of them I can get them pregnant thanks to this technique. And she was only doing natural matings, natural breeding. So could be an interesting tool to use in a kennel. So those techniques are good complements, and I think again, if we can use them in combination with a more accurate technique like progesterone assays, could be a very good way to use them to decrease the number of progesterone assays that are required to detect the day of ovulation. They are good complements, not as good as quantitative progesterone assays, but good way to estimate the day of ovulation. However, because it's more than an estimation, not a clear determination, this should be only used when you do fresh semen artificial insemination or fresh semen mating, because for, again, processed semen like shield semen or frozen semen, this will not be accurate enough to detect the day of ovulation. And I told you at the beginning that there is a very exciting field of veterinary medicine. It's based on those visualization techniques. Maybe you have heard about vaginal endoscopy, and if you attended to some of my lectures, when I touch on vaginal endoscopy, for me it's like playing PlayStation. I was raised with a PlayStation, and that's certainly why I love this technique, because it's really like playing video games. And the vaginal endoscopy allows us with a small camera to visualize the inside of the vagina of the bitch. And when we do that, this is what we can see. When we see this kind of morphology, this aspect of the vaginal wall, we know that the bitch is in an estrus, because it shows you that there is no specific modification. And look at here, this is a bitch in proestrus. We can see that the vaginal folds are bigger. They usually take a marshmallow-like morphology. This tells us that the bitch is in proestrus. If we see that, this very angulous shape of the vagina, we know that the bitch is in estrus. This is when we have the maximal conification of the vaginal cells, and this is when the vagina is the most resistant. This is usually the period which is around ovulation, if you remember. And when we see that, this pale aspect, we know that this is in diastrus. So based on the vaginal endoscopy, we can evaluate when, at which stage of the cycle the bitch is. To tell the truth, it's very close to vaginal smears. And you can wonder why this technique would be interesting. One reason is because of that. Look at this picture. If you see here, this is the cervix of the bitch, and you can see coming out of the cervix, you can see this purulent vulvar discharge. It's kind of weird thing coming out of the cervix. Look at the aspect of the vagina. This is a bitch with a chronic vaginitis and a chronic endometritis. And this is something we couldn't see with the vaginal smears because unfortunately we were not getting far enough and we were not able to collect those smears. When we did the endoscopy, because this was a bitch that was coming for infertility, we saw that. And we knew that something was wrong at the uterine level. So we had to take measures, specific treatments to try to fix this issue before breeding the bitch. Because in this situation, it was pointless breeding her because the sperm cell would have been killed because of the modification of the vaginal and uterine medium. And that was something we could only see thanks to endoscopy. So great technique that can allow us to see things we cannot detect with the vaginal smears. And now we can go real time, thanks to ovarian ultrasounds. And there's a story behind this. When I started at the vet school in 2005 in Alfort, near Paris, I spoke with my former boss. And he started 20 years ago in this field. And he told me that at this time, he was very interested in ovarian ultrasounds. And he knew that in women and males, they were doing ovarian ultrasounds to detect ovulation, which was something not difficult because when you think about it, in males, some follicles before ovulation are something between 3.5 to 6.5 centimeters in diameter, which means basically they are huge, so it's very easy to see them, even with very old ultrasound machines. In dogs, we are looking the follicles when they are on the ovaries, they are between 0.4 to 0.9 centimeter in diameters, very, very small. 20 years ago, the ultrasound machine we had were not good enough to clearly see what happens at the ovarian level during the estrus of the bitch. But one day he woke up and he said, maybe I should do my PhD thesis on this, on determination of ovulation in the bitch using ultrasound. And that's what he did. At this time, we had new ultrasound machine, which had a much better definition. And you will believe me on this one, but we could see the ovaries. This is an ovary in a bitch. And in fact, it can be you can see a clear white lining surrounding the ovary. This is the ovary here. We can detect it today with ultrasound. And in fact, this was an ovary in an oestrus in a bitch. When the bitch is in season, remember those big balls that are growing on the surface of the ovary that I showed you at the beginning? We can see them under ultrasound. Here is the ovary, and here are the ovarian follicles. So we can see them. We can measure them. We can follow them. So here, same thing, just before ovulation, you can see here a very nice aspect of the ovary with 5 follicles visible. Here they're big. They're big. They're 0.5 centimeter in diameter, and we can still detect them. And it can even tell us approximately the number. It can give us an idea of the number of oocytes that will be ovulated. For sure, we won't be able to tell you for sure, oh, this bitch is going to have 12, 14, 16, 22 puppies. But we can have an idea of the number, if it's potentially a large number of oocytes that are going to be ovulated, or a very small number. And this is what we see after ovulation. Here is the ovary, and you can see there are no ovarian follicles at all because ovulation occurred, those follicles ruptured, and the oocytes were released in the oviduct. So we don't see anything anymore on the ovary. This means that ovulation has occurred. And sometimes, if you're lucky, you can see this picture. Look here. This is the ovary. And if you look here, you have those black areas. And those black areas are in fact liquid, which has been released just after ovulation. When we see that, we can precisely time ovulation. We can be extremely accurate because this means that ovulation just occurred. Now, thanks to ovarian ultrasound, we can detect ovulation real time, and we can clearly say this is when ovulation occurred, this is when we should do the artificial insemination. And if we are dealing with an infertility case or frozen semen insemination, we can even time to do the insemination in the middle of the night if needed, because we know exactly when ovulation occurs. However, to do that, we need to do a follow-up, a little bit like the vaginal smears. We need to do several ultrasounds to see the follicular dynamics, to see how those follicles evolve, and to see when when ovulation occurs. So that's why usually this is not something we would do for fresh semen insemination or even child semen insemination, but this was mainly something we were reserving to frozen semen, because as I said before, frozen semen in certain cases it will live only maybe 4 hours in the genital tract of the bitch, and you need to be extremely accurate to know when to do the insemination to optimize the fertility and the prolificity of this bitch, because you know you're dealing with a very fragile semen, a highly processed semen. Thanks to ovarian ultrasound, we can be really, really accurate today. That's a great tool. I know that it's not always easy to access it, but when I was at the vet school, I was using ovarian ultrasound in all the bitches that were coming at the vet school, mainly because I had students and I wanted to show the students that it was easy. Many of them were telling me, oh, we cannot see anything with the ovaries, but in fact, we can. It's not a very difficult technique to learn, and if we practice a little bit, we are able to visualize those ovarian follicles and then to use this technique to detect ovulation. So great technique that can come in very handy, especially when you have issues with a progesterone machine during the weekend, for instance. But it's usually we were mainly using it for frozen semen or for infertility cases. So in conclusion, I showed you that there are plenty of different techniques available today to do timing of ovulation in the bitch. For sure, for me, hormonal assays are the gold standard, and they are the techniques with which we will have the best result when in terms of fertility. They are available in-house, and again, don't hesitate to discuss this with your veterinarian. There are techniques that can be performed in-house today, even if they are not doing a lot of canine reproduction. And we have other techniques that are available. I spoke about the vaginal smear, the resistivity of the vaginal mucus, etc. I would say, why not? This technique could be interesting, again, only for fresh semen or in combination with progesterone. But there are lots of variations, and the results you can obtain might vary from one bitch to the other. But there is one thing you guys can do. If you pick one of these techniques and you want to evaluate its accuracy, keep in mind that from ovulation to parturition, there is approximately 63 more than 1 days in the bitch. So if you know, you will see when the bitch gives birth to her puppies. So you can go back and determine the day of ovulation. And then if you see that the technique you use told you that this is when ovulation occurred, you know how accurate the technique is. So you can judge the accuracy of the technique by getting back to the day of ovulation after parturition in the bitch, if she was pregnant, obviously. But this is something you can evaluate, and you can then decide if you can use this technique or not for your breeding program. So we're done. We went through all of it. So thank you again for everybody for attending to this session tonight. It was always a pleasure. One thing I would like to add before we leave, this was a very interesting topic to prepare. And one thing important, I guess, is that keep in mind that when it comes to canine breeding, I had the opportunity to speak with breeders all across the world. And I realized that you guys usually share the same discussion, the same questions. So there are still lots of things to learn about canine reproduction, and I think it's important to be part of the discussion. So don't hesitate to join groups, breeders group. You can join our Elite Breeders Facebook group, be part of this discussion, because the more we share together, the more we can help this field moving forward, because this is the goal. Many things that were discovered in canine reproduction were done thanks to the partnership we had with breeders. And I really value this partnership. I did my PhD thanks to the partnership we had with breeders. And one of the things we really try to do now is really to be part of the conversation with you and to try to give you all the tools you need to properly manage and take the best from your breeding program. So again, thank you very much. Now we will do some Q&A. So Justin, I'll let you tell me the questions and we'll try to answer them. Thank you everybody, and thank you for joining tonight.